Hey guys, so in this video I'm going to be going over some of the problems from the Mendelian genetics lesson 7.2. Before you watch this, you should watch the introduction to Mendelian genetics video from Bozeman in part 1, and make sure you know terms like allele versus gene, dominant versus recessive alleles, a genotype, which is the genetic information, versus a phenotype, which is the physical outcome of that genetic information. Homologous, which refers to a purebred or an organism that has a genotype that is two of the same allele, whether it's dominant or recessive. And then heterozygous, which is an individual who is a hybrid or has two different alleles for a particular gene. And you should be able to differentiate between a monohybrid cross, which is for just one particular trait, and a dihybrid cross, which is examining organisms for two traits. We'll also do some work where we look at trihybrid conditions, but we won't have to draw out Punnett squares for that sort of thing. We'll also sort of learn about how Punnett squares help to demonstrate the concepts of the law of segregation and the law of independent assortment. So, in part 2, we're going to be looking at monohybrid crosses. There's a nice example in part 2 that shows a cross between two homozygotes that results in a heterozygous displaying the dominant trait, and then a cross between those two heterozygotes, which shows that in the F2 generation, you end up with 3:1 ratio in the phenotype and a 1:2:1 ratio in the genotype. And you can look at question number 1 under that part, which asks about indicating whether an organism is heterozygous, homozygous dominant, or homozygous recessive. And so letter A looks at two AAs, and because this genotype shows identical gene alleles, and those alleles are capitalized, we know this individual must be homozygous dominant. In other words, they're homozygous for the dominant allele. B shows a DD individual, and this one is pretty easy because there's two different traits. This would be a heterozygous individual, but we know that the dominant allele is going to be the one that actually shows up in simple Mendelian genetics. So this organism, even though they have a recessive allele here, the dominant allele would be the one that actually is expressed. C shows little I, and we know that those are lowercase, and so this person would be homozygous for the recessive. And then D is another example of a heterozygous. You can abbreviate these if you want. That's something that I do. Sometimes I just do HD, HT, and HR, or R. Sometimes, abbreviating these things helps me out. Let's quickly look at question 2. Question 2 is going to ask us for each of the genotypes below, determine the phenotype. Okay, so it's got RR, RR, and RR. And it tells us that round seeds are dominant to wrinkled. So our homozygous dominant individuals should definitely be round. And our homozygous recessive individuals should definitely be wrinkled. The only tricky question is, okay, what is this heterozygote going to show? And the dominant allele we know is round, and so our round allele would be the one that is expressed over the recessive. We also have the T's, TT, big T, little t, little t, little t. And then it tells us that bobtails are recessive and longtails are dominant. So this person is going to have a long tail, or this cat will have a long tail. This will be a bobtail, which is just a little short nub tail, and then the heterozygote will also display the long tail. So fairly easy for question 2. Question 3 is going to ask us to actually set up some Punnett squares. And so we want to set up a square for each of the crosses listed. So 3A is going to show a heterozygote individual crossed with a homozygous recessive individual. So I'm going to go ahead and set up my square over here. And then I'm going to put gametes for the female up here and gametes for the male up here. It doesn't really matter which is which in this case because this isn't a sex-linked gene, and it's just one that's pretty easy to figure out one way or another. I just like to split mine so that I do female on top and male on the side, or vice versa, just so I know what I'm doing. And for now, I'll just say the first one is female and the second one is male. Again, doesn't really matter. So the female alleles here can be capital R. So when we do meiosis, one of the daughter cells will get the capital R, and one of the daughter cells will get the little r. So I have 2 possible gametes coming from the female. The male, on the other hand, only has little r's. So both gametes that it produces is going to have little r's. And then we can do the cross and see that half of our offspring would be homozygous recessive, and half the offspring would be heterozygous, and then we would have a 50-50 split in the phenotypes as well. So it asks what percentage of the offspring will be round. I'm going to answer and say 50% of the offspring will be round in that case. B is going to say we're crossing 2 heterozygotes in this case. What percentage of these guys will be round? I'm going to go ahead and set up my square again. My female gametes are now going to be RR, and my gametes for male is also RR. We do my cross here. And now we have a bit of a different answer. So we're asking for round, and we know that from the previous question that round seeds are dominant to wrinkled. Which means that 75% of our offspring are going to be displaying round traits in this case, even though only one of them is actually homozygous dominant for the trait. Only 25% is homozygous dominant for the trait, and another 50% is heterozygous for the trait. C asks us to cross a dominant individual with a heterozygote. Put my capitals up here for the female, put a capital and a lowercase for the male. We do the cross. And now our answer is going to be 100% of our offspring will show the round trait. Question 4 is going to start to complicate things a little bit more. Question 4 is asking or stating that pea plants are crossed to demonstrate independent assortment. The traits are listed below. Capital letter is the dominant letter in all of the traits that are shown. So you should use that letter in your work. And then it gives you 4 possible conditions under which we're crossing. And I recommend using probability to determine the answers instead of drawing out the square, so you can see that probability is a useful way of getting the exact same answers that you need. And I'll show you sort of both side by side so that we get those answers. And they're telling us seed color, seed shape, flower color, plant height, pod shape. So all of the basic Mendel traits that he studied. So let's look at for problem 1. In a cross between 2 plants that are heterozygous for plant height, what is the probability the offspring will be heterozygous for this trait? Okay, so again, I'm going to draw up sort of my female and my male here, and it's telling us that they are heterozygous for plant height. And height in this case is tall versus dwarf, so we'll be doing T and T. So T for tall, and then lowercase t would imply that the plant is dwarf or short. what is the probability that the offspring will be heterozygous for this trait? So we can draw out the square if we wanted. I'll just sort of show you what that would look like, where we would have TT. But as we draw out these squares, it gets a little bit complicated to be able to do that. So we can also use probability to determine the same answer. So we're going to say, okay, what is the probability that this person, the female in this case, would send on one of the alleles or the other? So there's a 50-50 chance that this individual is going to send on a capital T, and a 50-50 chance that the other individual is going to send on a capital T or a lower T. So the chances in that case are the same. So the way we would do this is to look at the chances that the female is going to pass on a large T is 1 in 2, and the chances that the male is going to pass on a small t is 1 in 2. And we're going to multiply those, and so we would get a 1 in 4 chance that that could happen. And the 1 in 4 chance is actually, let's say this one down here. But we could have the odds that the female passes on a little t, which would be 1 in 2, and the male passes on a big T, which would be 1 in 2. And that would also be a 1 in 4 chance. That would be this example, right? And so we're going to have to add those up. And so we would get 1/4 plus 1/4, which would be 2/4, or a 50% chance of that occurrence. And if you look over here, we see, yeah, there's a 50% chance that that could occur. And so, I've used probability alongside the Punnett square to just sort of show this. But you can use just the probability. You don't need to use the square if you don't want to. For part 2 asks, if 2 white-flowered plants are crossed, what percentage of their offspring will be white-flowered? And so we have to look at flower color. It's going to be purple versus white. White is the recessive. So this would actually be little p times little p. I don't even need to do the probability here because there is 100% chance that both parents are going to pass on the little p. And so there is 100% chance that their offspring are going to be white-flowered. So that's the case of 2 purebred plants with the recessive allele passing on those recessive alleles to their offspring. A homozygous round-seeded plant is crossed with a homozygous wrinkle-seeded plant. What are the genotypes of the parents? Okay, this is fairly easy. Homozygous implies that the alleles are the same in the genes. And round is going to be dominant. So one of our parents is going to be homozygous dominant, showing the round. And the other parent is going to be homozygous but recessive, and so they are showing the wrinkled. What percentage of the offspring will also be homozygous? Okay, that's a good question. This parent is going to pass on only the capital R, and this parent is going to pass on only the little case r. And so 100% of the offspring are going to be heterozygous, which means 0 are also going to be homozygous. So when you cross 2 different purebred plants, they're going to result in a hybrid plant or a heterozygous plant in all cases. So that is also a fairly easy problem to do. And finally, let's look at 4. A plant with constricted pods is crossed with a plant that is heterozygous for inflated pods. What is the probability their offspring will have constricted pods? Okay, this is a little bit more complicated. And we can look and see that inflated is dominant to constricted. So one of our parents has constricted pods, so they must be little I, because they are not inflated, and they're showing constricted, which means they're recessive, homozygous recessive. And they are crossed with a plant that is heterozygous for inflated pods. So that would be an I, little I, right? The heterozygous individual. Like I said, again, I could draw out the square and show my parents and show the crosses. And I'll do that just really quick, just so you can see it. But I really want to solve this with probability. So I'm going to say, okay, there is a 1 in 1 chance that the female parent is passing on a little I, little I. And then there is a 50% chance that we are passing on the big I, right, which would give us a heterozygous individual. So this would be 1/2 or a 50% chance of having offspring with constricted pods, right? Constricted, we should be passing on the little I in this case. So that would be a 50% chance of having offspring with constricted pods. And if we go over here, we see yes. 50% of our offspring should have that particular condition. Question 5. Question 5 is a little bit different. This is referring to a test cross. So we're doing something completely different here. So a test cross is used to determine if the genotype of a plant with the dominant phenotype is homozygous or heterozygous by crossing it with a known homozygous recessive individual. Okay, so let's say you're out in the garden and you've grown a pea plant. You just put some seeds in the soil, you grew it, and it developed purple flowers. You don't know the genotype of that individual. Is it a homozygous dominant individual or is it a heterozygous individual? All we know is it's displaying the dominant trait, but we don't know its actual genetic makeup. So our question here is it this condition or is it this condition? So let's say that's our unknown female. And so to figure this information out, we are going to have to cross that plant with a known individual. The only known genotype that you can see physically in the phenotype is a recessive recessive individual. It's showing the recessive trait. It must be homozygous recessive. So we're going to cross it with little p, this guy over here. Now, if the unknown is homozygous, all of the offspring of the test cross have a what type of phenotype? If the unknown is heterozygous, half of the offspring will have a what phenotype? This is a bit of a tricky question. Again, we can do it with our Punnett squares. If we want, we can say, okay, the female is giving us a dominant, the male is giving us a recessive. We're going to end up with 100% dominant trait being shown in the phenotype of the offspring. Or we have this other condition, right? We have this guy where the female is heterozygous and only half or 50% are showing the dominant phenotype. So, we have the difference there. Again, you can solve this using probability instead to say that, okay, if this is, in one case versus two, in case number 1, we have a 1 in 1 chance of passing on the dominant from the female, and we have a 0 chance of passing on the dominant from the male, and so there would be a 0 chance of recessive and 100% chance of dominant. Versus the other part, we could do, okay, they have a half a chance to send on the dominant, and then a 0% chance over here of sending on the dominant, and half versus 1. Which means we are going to have half showing the dominant trait. So you can use probability to do the same thing as the Punnett square. Sorry, question 6 is going to give us an actual scenario where we have to apply this test cross. And she in the example, we end up actually seeing information being provided to us in terms of data. So a breeder has a black male border collie that has won numerous awards. The breeder would like to use the dog for breeding if he is purebred. So here's the male over here. Is he BB or is he B little b? We just don't know yet. And to learn this information, she's going to test cross him with the female who's little b, little b. Okay, she ends up with a litter of 9 puppies, 4 of which are black and 5 of which are red. What would you advise the breeder? I am going to assume that because a recessive trait is being seen in the offspring, that at least some of the offspring must be displaying the homozygous recessive genotype, and that means that the father must have passed on a recessive allele to some of his offspring. So we can set the square up. We can say that if, if this is the female, if this is the female and this is the male, then we're able to see that some of the offspring will display red and some of the offspring will display black. And so I would say to this breeder, no, that your male dog is not a purebred for this trait, and instead you may, or actually none of the black puppies would be purebred for that trait either, and so you wouldn't want to use this dog for showing. A little bit of a different trait between black hair and white hair versus long hair versus short hair, but essentially it comes to the same thing. I'm going to draw out the full square of this just because I can, but this is data that you can figure out through probability. You don't need to draw out the square every time. So our female versus our male gametes. In this case, one of the parents, let's say the female in this case, is purebred black and purebred long hair, and she's being crossed with a male who is purebred white and purebred short hair. And so the only alleles that she can produce in gametes are going to be BL, and the only gametes he can produce are little b, little l. And so those would come together into new offspring. I'll make this more distinctive for you. And, you know, here's our parent generation. Here's our F1 generation. What is our F2 generation going to look like? So we have to figure out in this case, okay, what possible gametes can be sent on from the individuals in the F1 generation? Well, some of their gametes could be this B and this L, right? Some of their gametes could be this B and this L. Some of their gametes could be capital B, lower L. And some of their gametes could be little b, little l. So we have 4 possible gametes that can get passed on to the F2 generation. And that's going to require a complex Punnett square. And we can assume that both our male and our female for this cross is going to be heterozygous and going to be producing these gametes. So for the female, I would write the first gamete, the second gamete, the third gamete, and the fourth gamete. And I'm going to do the same for the male. We're going to basically put it going down. And now we want to cross them and we want to see what sort of the outcomes are. So I'm going to do the B's first because for me it's easier to just cross the one letter at a time. It's up to you. You can do it however you want, but for me that's easier. Okay, I've done my B's, and now I'm going to do the L's. So then you want to know, okay, how many of each phenotype? This is a question. You can count them out. I always start in the lower right-hand corner, because for some reason that's easier for me to say, okay, one individual is little b, little l, so we're going to have 1 out of 16 displaying the white short trait. How many other individuals are displaying the short trait? Only these 3 are displaying the short trait. So that's 3 out of 16. And all of them are black short. How many individuals are displaying the white trait? Here's 1, here's 2, here's 3. So 1, 2, 3. So that's 3 out of 16 that are white, but they're all showing long. And then we have these guys, and these 2 guys. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16 displaying the black long trait. And so this ratio should be familiar to you, 9 to 3 to 3 to 1, which is sort of a classic ratio when you're looking at the dihybrid cross. Question 2 here is going to ask for a cross between 2 slightly different parents. So in this case, let's say our female and our male are going to be B, B, L, L, and B, B, L, L. So we want to cross them to figure out how many of the offspring are black and short, black and long, white and short, white and long. And we can do this using the Punnett square. But we may not have to use the full Punnett square. What we could do is we could draw out a Punnett square, or we could use probability to determine these answers. For number 2 and 3, I think I'll just draw out the square for you guys, and then for 4 and 5, I'll show you how to use probability. So in this case, what gametes can be produced? This female can produce 4 gametes, 4 possible gametes. But the male can only produce 2 possible gametes. So what I'm going to do is I'm actually going to cross these in a slightly different square where I draw my female on the top and my male on the side. Okay, so I have these 8 possible outcomes, and my I'm going to have to add up basically what my conditions are. So let's see, I have 1 who's going to be 1 out of 8 who's going to show the white short traits. Let's look at how many other individuals are, oh, I made a mistake, I just caught it. Let's look at how many other individuals are showing white traits. It's going to be these 3. 3 out of 8 that are white but long. And then we have everyone here showing black but short. And everyone here showing black long. So our ratio is 3 to 1 to 3 to 1, or sometimes people order them 3 to 3 to 1 to 1. But that's what we have for step 2. In question 3, we have a parents who are big B, little l, and little b, big L, big L. So what sort of gametes can these parents make? This parent can only make one type of gamete, so that's fairly straightforward. The female can make 2 types of gametes, so that's going to be pretty straightforward. And I can just sort of show a single cross if I want to for the male, right? He's showing only this gamete. Actually, I don't even need the other half here. I really just need this. And the female is showing big B, little l, and little b, little l. So, 50% or 1/2 are going to show black long, and 1/2 or 50% are showing white long. So you could say this is a 1-to-1 ratio, a 2-to-2 ratio, whatever you want to say it is, but fairly straightforward in this case. Okay, let's use some probability to do part 4. Using probability, not a Punnett square, what would the odds be of having a white short-haired offspring from parents who are heterozygous in this case, and heterozygous for one trait and dominant for the other? Okay. What is the odds of having a white short-haired offspring from these parents? So, white offspring are going to inherit little b, right? Because we know that they must be homozygous recessive. So there is a 1 in 2 chance of inheriting little b from the female, and a 1 in 2 chance of inheriting little b from the male. So we do 1 in 2, 1 in 2 is going to get us to 1 in 4 chance that we get the little b conditions. Now, what is the chance that they're going to be short-haired? The female can pass on a short-haired allele, but the male cannot. So there's a 0% chance in that case. So this is going to be 1 in 4 times 0. 4 times 0, which would be 0 chance of that happening. So it's pretty straightforward, and I figured that out just through probability. Didn't even have to do the actual cross out. Very, very, very easy. If I had, I would have figured it out by looking at the whole square, but I didn't need to. So that's number 4. 5 is going to be more complicated. The breeder now wants to predict eye color in their bred offspring. Eye color is either red or brown. What would the odds be of having brown-eyed, white, long-haired offspring from parents who are, let's say the female is BB, L, L, R, R, and the male is BB, L, L, R, R. Okay. So this is a little bit tricky. So we want to know, okay, what are the odds of having a brown-eyed? So let's start with brown eyes first. Brown-eyed, let's see, red is dominant, brown is recessive. So there is a 1/2 chance that this female will pass on the lowercase r, and there's a 1/2 chance that this male will pass on the lowercase r. So 1/2, 1/2, 1/4. So that's one of our values. What are the odds of having a white long-haired offspring? Okay, white is going to be capital B. So there's a 1/2 chance here and a 100% or 1 chance here. So in that case, it's going to be 1/2 times 1, 1/2. So let's do this eye, coat, color. And then it's asking for long-haired. Okay. There's a 100% chance that the female will, but only 1/2 that the male will. So now we can use all of these factors to figure it out. Now we would multiply all these up, and you would get a 1 in 16th chance of having the brown-eyed long-haired, white-colored individual. So easy way to do a tri-hybrid cross there.